Yeah, if my voice permits, I'll tell you a bit about one of my postdoc projects here at Lund University that I'm working on together with Eric Svensson. And I changed the title a little bit. There was using deep learning at the end, but I took it away because I'm actually not going to talk so much about it, but I really want to pick your, or get your opinion on what to do with the data, what kinds of data we can extract from organisms and parts of organisms. So I'm really interested in color polymorphisms, which is within species, or yeah, within species variation in color traits. And there are many types of polymorphisms. For example, our blood group is one. There are other types of polymorphisms in morphology, absolute color, color pattern, etc. And it's a good system to study ecological and especially evolutionary dynamics, because they're all based on some discrete genetic mechanism, right? So we can get discrete or continuous phenotypic variation based on a discrete genetic mechanism. And it's as you might imagine, it's of course a fantastic system, especially in the context of computer vision, because it's very visual. So in Eric's lab, we use the Ischnura elegans system, which is a trimorphic, female-limited color polymorphism. So there are three female morphs, and it's controlled by a single locus, so three alleles in a dominance hierarchy, and the males are monomorphic. This is a male mimicking morph, the A morph, and then two gynochrome morphs, so the female-like morphs, I and O morph. And it's thought that this male mimicking morph has evolved in response to male mating harassment, which can have fitness consequences. So there has been this evolution into a male-looking phenotype to avoid this harassment, which can often result in death. So this is a different species, but we see this a lot in the field in Ischnura elegans, that females are actually drowning or dying from aggressive mating attempts. And Eric's lab has been very successful in showing that this is actually quite a common thing in populations around Lund, but it's also remarkably stable. So if you look at the male mimic, not only is it, these are the morph frequencies in different population. Eric's been following 16 populations around Lund. It's incredibly stable over time, the frequencies, right? So, and in addition, it's also unexpected that the male mimicking morph, which is the blue line, occurs at such high frequencies. So you could, as you would imagine, if something is very frequent within a population, the males easily form a search image and then target it. So it's actually counterintuitive that the mimics have the highest frequency of them all, like up to 80%. So these are the questions that I'm after. On the one hand, how discrete or continuous is this polymorphism along dimensions of color, but also shape? So we do have observed that there is some covariance with shape traits, between shape and color traits. And how strong, and then asking how strongly are these dimensions of variation related to mate choice, mating success, and fecundity, and then how stable, in comparison to these frequencies, how stable is the morphospace occupancy through time? And there are a whole range of other really interesting questions that I don't have time to get into today, like ontogenetic aspects or population-specific dynamics. So it's a really fascinating system. And yeah, I was able to use Eric's fantastic database, which is now amounts to 20,000 imaged specimen. There are way more recorded specimen, I think now just over 70,000. But 20,000 of them have these highly standardized image data. And we use, I think Quentin brought this up, a flatbed scanner. So that's as standardized as you can get. The lighting is always the same, contrast, color space, etc. So it's a really nice way to take lots of images really fast. And so every image we can then connect to a record of other information, such as fecundity, so clutch size, copulation status. We even have amazing things like parasite count, like individual mites, ectoparasites. and other relevant metadata. And now I'm happy to make it even so that it's become even simpler. So we've played around, or we've experimented with these QR codes, which is a thing that I would in general suggest if you, or would propose if you use field data or lab data. It's a really nice way to read in information that is encoded in your images. So that's really, instead of manually, connecting an image name to a number in your database, the QR recognition is very robust. I mean, it's a very established computer vision mechanism, and in Python it's really easy to generate these tiny QR codes. So yeah, that's happening now. And for the approach, we're using Phenotypic, which is a package that I've been working on for a long time already during my PhD, and that got published a few years ago, which is, as Ben pointed out, it's a system. It's a system to do computer vision. So it takes care of data management, labeling, annotation, but then later, once you've trained a model, that capacity is not in Phenotypic, but any model that you've trained, you can tie back into Phenotypic and replace the manual labeling step with an automatic one. So it's very modular in what it can do. And then it also does the trait extraction. And so for this project, I used a very shallow model. Now I'm a bit less secure after yesterday talking about accuracies, but in our test data set, the accuracy is 98%, which sounds good, but in fact there's still a lot of manual work left to do. So 98% of the pixels were predicted correctly, but if you're interested in fine details, there's still quite a bit of variation that is left and that we have to manually fix, which is also something how automatic are our automatic pipelines? It's a bit of a euphemism, right? I guess we saw with Yuchen's work that you have to go in and you have to fix little things here and there. So it's never really fully automatic to a really high accuracy. And this is not the deep learning pipeline, but this is just a GIF of the annotating the training data using signal processing. So instead of using deep learning to scale up, because these images are so highly standardized, we just do this sort of a bit clumsy approach to segment out the different body parts and then feed it into the model. And because these images are so highly standardized, and this is just the model for the entire outer shape, training, it's very, the model, the loss function reaches its minimum fairly soon. So this is just a random, this is not the actual pipeline or the actual training in the end, it's just an animation to show how quickly it converges to something useful. So what do we get out of it? I'm interested in not only the whole body, but in different body parts and how they vary either individually or together. And to do so, I extract both shape traits, and these are just really generic descriptors of area, diameter, but also circularity. So these, the head and the thorax and the pronotum are very circular, so they have a value close to 1. The abdomen is not very circular, it has a value close to 0. So that sort of theta. And also color pattern traits, which I borrowed from a package that is called PyRadiomics, which was developed for medical image analysis, which by many traits that I don't understand, they're very mathy, very abstract, but they together, they have the capacity to essentially map out phenotypic variation in texture in morphospace. And again, just by itself, these are some of the metrics. By themselves, they're not really meaningful. I mean, some are, right? Like the diameter can tell you something about variation in different body parts. I believe this is thorax, and the males are just slightly smaller. So some of the information is useful, other is not. And for me, it starts to become interesting when I map them out all together. So these are, this is information from all 3 color channels, and it includes 36 texture traits, and I believe 7 shape traits. And each dot is an individual, and the different ellipsoids are the different morphotypes. And the first axis of this morphospace is mostly variation that's coming from shape features, and the second axis is coming from shape features. And because we use computer vision, we can actually map the extracted masks back onto the space. And you can see that, for example, here, down there, it's continuous, right? So it's not very discrete as you would imagine it, but it's a continuum of mostly of shape, right? Because PC2 is mostly the shape axis. Yeah, so it's because the whole body in damselfly is a bit difficult to compare. We can also do it just for the thorax, and there I think it becomes nicely visible. And what's interesting is that the separation among morphs mostly occurs the shape traits. So it's just, as I said, the males are smaller and more slender, and the male mimics are also smaller and more slender. So it's not just color, it's also shape. However, and that is something that I also thought was really interesting, is this variation in the texture axis. So on PC1, and these are just the centroids for the different years. Between years, there's massive variation among textures. I don't know yet what that means. It could mean that texture or coloration is more flexible than shape. Shape is more conserved. And yeah, I'm looking forward to get more data to solidify this finding. We can, of course, also do this for the different body parts, and because this is actually not a PCA, it's a multiple factor analysis. So it recognizes, or it accounts for different parts of the body that I put in, and then gives me this global analysis, but also separate ones. And I think I'm running a bit out of time.
I'm trying to be a little bit intimidating.
Okay, no, that's good. That's good. I'll speed up a bit so we have time for a few questions. As I said, the next steps, and this is just to show what I'm doing right now. I'm now trying to map population probability, and these are fits from a generalized additive model onto this morphospace. And as we can see, there are different mating optima. But overall, it seems like the female-like morphs are more attractive than the male-like morphs, which is interesting, I think, given the high frequency of the male morphs. And also, the male-like morphs tend to have smaller clutches, at least on some axes, right? So this is the logged clutch size, because there's a really big spread in the number of eggs between something a few dozen and several hundred. Then I think it matches up nicely with the copulation status. And Dario is now collecting more images to get the remaining years using phenotypic and the trained model. And I think we're making good progress with the system. So some conclusions. There is some overlap between the morphs, but otherwise good separation, mostly along shape. There's higher copulation probability and fecundity if morphs are more female-like and less male-like. So this is my research program that I'm interested in, and people like Masahito that I'm talking a lot about with Masahito and Eric. And in mapping out phenomes, I'm more in this corner. So I'm more comfortable that I have lots of individuals that I each individually don't map out very deeply, because I'm interested in these broader population level or time series patterns. Yeah, thanks for listening.